Hello everyone, I am Dr. Pooja Johari and today I will be discussing the NEET PG Recall 2022 of the subject Biochemistry. So overall, if I have to give rating for the questions asked this year, I found the questions to be relatively simple. I mean, there was no complication involved as such. There were some straightforward questions. One or two questions were from molecular biology and some of the questions were from general biochemistry. Right. So I hope you people did a pretty good job in your exam. So let's start up with the discussion of the questions. Let's see question number 1. DNA packing is done by. Now see, this is a very interesting question and a very important question. This I have been discussing about the packaging of DNA in my previous lectures also. Always remember DNA is actually packed with histones. Histones, which are the basic proteins rich in lysine and arginine. Because all of us know that DNA is negatively charged, right? And histones having basic proteins means they have an extra amino group or they have an extra positive charge. So these histones are actually 4 in number and they are present in double like H2A, H2B, H3 and H4. So it is present as the octamer 4, 5, 6, 7, 8. And DNA is wrapped like this. It's like it resembles the NCERT symbol. Okay. And this entire thing is known as a nucleosome. So nucleosome is basically DNA plus histones. And the most important function of histones is they regulate gene expression. They regulate the process of transcription. Right. So DNA packing is done by histones, which are the basic proteins rich in lysine and arginine. And overall, it is known as a nucleosome, which gives a beads on a string appearance. Okay. So the answer will be this. Moving on to the next question. The following histopathological images seen in the patients. Now see, whenever these image-based questions come, right? This is, you can see a crumpled type of experience, a crumpled type of appearance is there. And this I have discussed in detail in my video on sphingolipidosis. This is characteristic feature of Gaucher's disease. And you can see findings were anemia, thrombocytopenia, and bony pain. And always remember, when a person is having Gaucher's disease, there is Erlenmeyer type of deformity. And all of us know that Gaucher's disease is due to the deficiency of beta-glucose. This is a very, very important, very common lysosomal storage disorder. Let's look at what are the biochemical manifestations of this. Deficient enzyme is glucocerebrosidase. The most important feature about again sphingolipidosis is no very red spot is there in the macula. Right? Bone crisis with severe pain and swelling can occur. Bony pain. And if we carry out the X-ray of the femur, we can see that it resembles that Erlenmeyer flask kind of thing. It's like this. Right? And no mental deterioration is visible. So these are the basic clinical manifestations of Gaucher's disease. And the most important one is bone marrow examination. You can see that they are actually known as Gaucher cells. The wrinkled paper appearance resulting from the presence of intracytoplasmic substrate inclusions. And we know Gaucher's is due to the deficiency of beta-glucocerebrosidase. So the answer will be this. Sphingomyelinase deficiency leads to Niemann-Pick disease. Glucose 6-phosphatase leads to von Gierke's disease, and alpha-glucocerebrosidase is altogether different. These are all different types of sphingolipidosis. Moving on to the third question. An alcoholic patient on anti-tubercular drugs for the past 3 months complains of numbness, tingling, and burning sensation in all the extremities. Which vitamin should be deficient? Now see, here 2 important features are given. One is an alcoholic patient and on anti-tuberculosis drugs. So one drug which comes to our mind is isoniazid. Whenever we talk about isoniazid, it is related with B6. B6, which is pyridoxine or pyridoxal phosphate. B6, which is also related to B3. But here, there is a slight twist. One is B6 is not given, so B6 is ruled out. But here we are talking about the alcoholic patient. And we can say that numbness, tingling, and burning sensation is there. So whenever we talk about alcoholism, the most important vitamin which is related to alcoholic metabolism is B1 or thiamine. Right, because this thiamine is actually responsible for the conversion of pyruvic acid to acetyl coenzyme. And deficiency of this leads to beriberi. So here B6, B3 is not given. So the answer will be thiamine, pyrophosphate or thiamine. If it wasn't mentioned alcoholic patient and if it was mentioned B6, the preference would have been given to B6. Clear? And moving on to the next question. A paint factory factory worker working since 20 years complains of insomnia, fatigue, headache, constipation, and joint pain. Is found to have anemia. Now here we have to gather just two important points. One is second is anemia. So obviously paint factory worker is there who's working in the paint one. So it has something to do with lead poisoning. And here it is clear that he is suffering from anemia also. That means it is related to the synthesis of heme. So always remember, all of us have, I have discussed the synthesis of heme in my previous videos also. Whenever we talk about heme synthesis, lead poisoning, it always affects two enzymes. One is this pyrochelatase, which is the last enzyme leading to the synthesis of heme, and another is PPG, prophylinogen synthase. Now, since this enzyme is not mentioned here, the answer has to be okay. ALA synthase is the rate limiting enzyme of heme synthesis. So the answer to this question will be pyrochelatase. Clear? Moving on to the next question. Enzyme defect in McArdle's disease. Now, always remember McArdle's disease is a very, very important glycogen storage disorder. And if we see, so you see, you can say that it's like wo physics, chemistry or maths mein hoshyar tha. This is a mnemonic to remember the glycogen storage diseases. Out of this, PMP are related to muscle storage. P stands for Pom's disease, M stands for McArdle's disease, and T stands for Taruy's disease. So here we can say McArdle's, the deficient enzyme has to be muscle, so myophysis. And this is a very, very important point, a point of differentiation also between McArdle's disease and Taruy's disease. There is a second wind phenomenon present in patients suffering from McArdle's disease. Means if a person relaxes for some time, after that he can regain the activity. But in Taruy's disease, it is also muscle related disorder. The deficient enzyme is PFK, but no second wind phenomenon is present. And if the patient suffering from Taruy's disease is given a diet rich in carbohydrates, the condition becomes more severe. But here it is simple. The answer will be myophosphorylis or muscle phosphorylase. The next question, cherry red spots. Now see, whenever you talk about cherry red spots, okay, always remember this is a characteristic feature of sphingolipidosis. Right, sphingolipidosis means the alcohol is sphignol and they are a very, very important class of phospholipids. And cherry red spot, if we compare this, is a characteristic feature of in Gaucher's disease, no cherry red formation is there. Instead, it is like pseudo. Similarly, in Thames, no cherry red spots formation and mental retardation is seen. And Hunter syndrome is altogether different. Hunter syndrome is a mucopolysaccharidosis. Right? So the answer will be T6 disease. And I have prepared a very interesting mnemonic on T6. I'll just show it to you. See. But first of all, cherry red spot in the macula is seen in all except Gaucher's, Fabry's, and Krabbe's disease. Except these, other sphingolipidosis have a characteristic cherry red spot formation in the macula. This is a very interesting mnemonic. T6 testing obviously is recommended. It is autosomal recessive macula. And it was first discovered in Ashkenazi Jews. And it leads to CNS degeneration. The deficient enzyme is hexosaminidase A. And it's obviously a lysosomal storage disorder. So the answer will be Tay-Sachs disease. Moving on to the next question. Inhibitor of complex 5. A very interesting question. All of us know that EPC has basically 5 complexes. Complex 1, 2, 3, and 4. Here they are talking about complex 4. So the major inhibitors of complex 4 are carbon monoxide, obviously cyanide, and sodium acid. These are the 3 very, very important inhibitors of complex 4, which is cytochrome A1, A3 oxidase. So the answer will be cyanide. Not carbon dioxide, it is carbon monoxide. Oligomycin is of complex 5. This is of complex 2 or 3. Antimycin. Because there are 4 complexes, 1, 2, 3, and along with 5, which leads to the synthesis of. Correct? So the answer will be cyanide. Moving on to the next question. FAD status. FAD is vitamin D, or it is riboflavin. Always remember, whenever we have to specify, we have to check the status of riboflavin, we always estimate the assay of glutathione reductase, a very, very important enzyme which functions as an antioxidant also. Glutathione, which is actually a tripeptide formed of 3 amino acids. And whenever we have thymine, we have to carry out transketolase. Because thymine, TPP, is an important component of transketolase, which is of HMP pathway. Moving on to the next question. True about Barbars effect. Now always remember, Barbars effect is actually aerobic glycolysis with lactate formation. It always increases the uptake of glucose. This we have already discussed in, I have discussed it in my videos on glycolysis, and in which I have discussed Pasteur effect and Barbars effect. Now I'll just show you what are the important features. You see that cancer cells take large amount of glucose, form lactic acid even in the presence of oxygen. So basically it is aerobic glycolysis. Normal cell source of ATP is oxidative phosphorylation. Now when we talk about cancerous cells, increased glucose uptake by cancer cells actually helps us in scanning this PET scan, basically to detect the tumors. So this is actually a very, very important process. You can say pathway occurring in the cancerous cells. The next, when a codon is changed into a stop codon, very simple, it leads to nonsense mutation. Not missense, not silent, not stop codon. Because missense is something else in which one base is replaced by another. Silent mutation is representing wobble hypothesis. Mutation is altogether different. So this is nonsense mutation when UGA, UAG, or the nonsense codons appear on the way. Moving on to the next question. A Mediterranean person didn't receive primaquine. Which pathway is affected by G6PDH deficiency? Now see, primaquine, anti-malarial drug, right? Whenever this anti-malarial drug, and it is written here, which pathway is affected by G6PDH deficiency? Only one, pathway, a shunt comes into our mind, HMP. Because HMP is the shunt which generates NADPH with the help of enzyme G6PDH, which is the rate limiting enzyme also. And basically it is related to oxidative stress. So the answer is relatively very easy. It is HMP pathway because deficiency of G6PDH leads to hemolytic anemia. So hemolysis is there, splitting of heme is there, right? Condition shown below is due to which vitamin deficiency? Now, simple, we can see the white spots here as it is clearly visible. And whenever we talk about white hot spots, only one vitamin comes into our mind, which plays a very, very important role in visual cycle. So it will be vitamin A, which leads to the formation of white hot spots. True about telomeres and telomerase. Now see, always remember telomeres. How do we define telomeres? They are actually the set of repetitive DNA sequences at the end of a chromosome. So whenever a cell divides, the telomere starts becoming slightly shorter. And eventually they become so short with successive divisions, the cell can no longer divide successfully and the cell dies. And telomerase is the enzyme which is responsible for the working of telomeres. So if we see what can be true, there will be gradual decreasing length of telomere with aging. So this will be the answer. And always remember they are present in the stem cells and they are present in the germinal cells, not in somatic cells. They are non-coding sequences and this. Clear? Rice water diarrhea intoxication is due to which ganglioside? Again, it is simple GM1 ganglioside receptor. Then mechanism of action of botulinum toxin is, it is related, actually it is nothing but it is tetanus toxin only. So the major function catalyzed by this is it always inhibits the release of acetylcholine, always from the lower cortex. And if you look at the mechanism of action of TT, it's like this. It always inhibits the release. Then transport of fatty acid through mitochondrial membrane is why. Always remember it has to be because the oxidation takes place in the mitochondria and the synthesis of fatty acid takes place in the cytosol. So from the cytosol, fatty acid has to be transported to the mitochondria. So how it is carried? It is carried by carnitine shuttle, which is a derivative of lysine. Right? Then LC80 is altogether different. LC80 is lecithin cholesterol acyl transferase, which is responsible for the synthesis of cholesterol. Cholesterol transporter is different. So the answer will be carnitine. Now, which of the following has autosomal pattern of inheritance? Now, this again is a very interesting question. Cystic fibrosis, it is having autosomal pattern of, it is actually autosomal recessive pattern of inheritance. The rest are autosomal dominant. Autosomal. Then a woman on antidepressant presents with bleeding, she gives a history of bulky stools which stick to the pan. Which of the following deficiency can cause bleeding in this condition? Now see, on antidepressants present with bleeding. Whenever this bleeding concept, because it is a case of steatorrhea. Which of the following deficiency can cause bleeding? It is vitamin K, because vitamin K is responsible for the post-translational modification of the clotting factors 2, 7, 9, and 10. 7, 9, and right? Because all are fat soluble vitamins. Clear? And bulky spores, which stick to the pan, characteristic of Steatorrhea only, right? Then identify the acid-base disorder in the patient with the following values. Now you can see pH 7.2 and PCOT is 80. The normal value of this is 45 is so high, and pH is slightly going towards acidic side. So when pH is acidic, PCO2 is high, it is a case of respiratory disease, as you can see here. Respiratory acidosis will give this finding, metabolic acidosis will give this finding, alkalosis will give this finding, and metabolic will give this finding. So this was a review from my side to all questions of biochemistry which were asked in your PG exam. So wishing you all the best for your results. I hope this year you people clear it. So thank you so much for listening patiently to my video. All the best. Take care.